Is my screen visible?
Yes, it is.
Okay, so good evening, everyone. I'm Anshika, and today I'll be presenting on the topic fingerprints and their role in forensics. So, today we'll be discussing the type of fingerprints, reason for occurrence of these fingerprints, and their role in forensics. And we'll be also looking at some other aspects along with these. So, before we start, just a second. Yes. So, starting with the definition of fingerprints, fingerprints are defined as a mask formed by the patterns on the fingertips. Now, the question is how are they transferred to surfaces and how do they form their impressions? So, our fingerprints have different patterns of ridges and furrows over them. If you look at your fingers, you can very clearly see some rounded or some square or some different kind of shapes on your fingertips, as you can also see in the diagram on the right hand side. So our fingerprints have different patterns of ridges and furrows, and these ridges have micropores, which perspire upon contact with surfaces. Due to this perspiration, an impression is formed on the surface of contact. Every person has a unique set of fingerprints, which doesn't match even within the fingers. So if we look closely, a confusion on what was the need of fingerprints and why did they evolve comes forward. Upon compilation of data from various sources, Henry Lee has provided three reasons for evolution of fingerprints. The first one being occurrence of perspiration. Fingerprints act as an outlet for various oils and sweat. So this is the first reason why fingerprints evolved. Second reason being tactile facility. So it has been argued by scholars that fingertips and the ridges and furrow pattern provides a higher perception of touch as compared to other surfaces of a body. However, as a counterpart to this study, a study showed that ridge and furrow pattern provides lesser surface area for contact if we touch a surface. The third reason for fingerprint evolution is supposed to be ease in gripping surfaces. The third reason for fingerprint evolution is supposed to be ease in gripping surfaces. It gains support from the fact that animals like koala and some arboreal primates also have fingerprints that aid in gripping surfaces. This also gains support from the observation that if we dip our hands in water for some time, the ridges swell out. And you can very easily observe that if with those swollen ridges, if you try to pick something up, you automatically gain a better grip. So this observation was then compared to other surfaces on our skin. So it was concluded that the ridges that we have on our fingertips are providing us with a better opportunity and a better gripping of whatever we are trying to take a grasp of. Now, if we have to talk about types of fingerprints, before that we'll have to look into types of ridges and furrows. So on the left hand side, you see 7 basic patterns of ridges, and on the right hand side are their respective names. So these patterns then convolute and form much complex patterns which lead into fingerprints. So suppose that you have a crossover which is followed by a spur and then which is followed by a ridge ending. So these kind of patterns convolute together and then they form a much complex type of fingerprint. This classification of ridges and furrows was given by John Berry. And according to this, we have 3 basic type of fingerprints: arcs, loops, and holes. Another categorization of composite fingerprints has also been provided by some scholars. While we have some scholars which argue that composite fingerprints, which comprise of twin loop and lateral pocket loop, are nothing but a subcategory of loops. So if you see at the diagram in the next slide over here, this is the classification of fingerprints into 3 main parts and their sub-classification into types like simple, tended, ulnar, radial, concentric, single spiral, and double spiral. So here is a diagram. This is a simple arch. This one is a tented arch. This is a left loop and this is a right loop. So yeah, so ulnar and radial loops are also known as left and right loop, depending on what is the position and what hand we are seeing for it. So as we all know that our radius is towards our thumb side and our ulna is towards our little finger side. So if we are seeing our left hand and the position of the loop is going towards left side, so we automatically know that it is going towards our thumb. So we know that this one is our radial loop. Similarly, we have hold or simple hold. Then we have single spiral holds, double spiral holds, and then we have this twin loop and lateral pocket loop. So now, what are the characteristics of identification of fingerprints? So we very easily classified these into arcs, holes, loops, and all. But what are the things that we take into consideration when we are saying that this is an arc or this is a hole? So we firstly look at pattern type, then we look at the ridge counting, then we look at the ridge characteristics, that how many times does it go in circles, what kind of structures it is forming, and then we look at the number of loops and tents. So now we'll be moving into the next section, which is the role of fingerprints in forensics. Whenever we have an ongoing forensic or criminal investigation, one of the bases of collection of fingerprints, we have three categories. First one is latent prints, second is visible prints, and third is plastic prints. So latent prints are the one that we pick up from the crime scene. So you might have seen it in many shows that they go around with a brush and they just put some black colored powder on surfaces and then they pick up the fingerprint using a tape or a surface like that. So when we are collecting fingerprints from a crime scene or some specific site, that is known as latent prints. Visible prints are basically the prints which we give with complete knowledge. So just for example, you're giving your fingerprint to your phone so that you can unlock it. So that is a visible print. And plastic prints refers to 3D fingerprints. Like you might have also seen in some movies that they put paint or some wax kind of stuff on the fingers and then they just peel it off and the entire design of fingerprints just comes onto the surface of the tape or paint they were using. So those are the three-dimensional plastic prints. Then there are these two types of surfaces, porous surface and non-porous surface. The glass-like surface, which we have, or the surface of a knife, they are the non-porous surfaces. And anything other than such kind of surfaces like paper, cardboards, etc., all of them classify as porous surfaces. Now, this is the role of fingerprints in forensics. The first one being criminal investigation. So what basically happens is that we collect fingerprints from the crime site. Then we take into consideration all of the accused people, and then we compare the fingerprints obtained from the site and the fingerprints of those people. And we look for similarities basically. And as I've mentioned before, fingerprints are very unique, so no two fingerprints will match. Even the fingerprint of your one finger won't match with the fingerprint of another finger. So now the next role of fingerprints in forensics, detection of drug use. So as I told, the ridges on the fingertips have micropores and they occasionally secrete out trace amounts of drugs if we have taken any. So along with the oils and sweats that's coming from these micropores, trace amounts of drugs can also be traced with the help of these fingerprints. The third application is medical condition diagnosis. Now we have some medical conditions like the Down syndrome, in which the third and the fourth finger of each of the hand doesn't have fingerprints or has very highly diminished fingerprints. So you can't basically see if the person has fingerprints or not. Similarly, we have Turner syndrome and other such medical conditions where you can diagnose the medical condition just by looking at the fingertips. This has also been seen in the case of Alzheimer, but I'm not pretty sure about what leads into the conclusion that the person has Alzheimer just on the basis of looking at their fingerprints. Now, at last, the fourth application is the construction of a forensic database. So we are looking at the victim, we are looking at the accused people, we are taking their fingerprints and we are mentioning it in a database. So in case we have a similar kind of related crime occurring, so we can just compare the data we have with the data we will get at the time of that case. So that would help us in easier investigations. So that is why we have forensic databases. So now if we look at the methods to develop fingerprints, it means that, okay, we understood that we have fingerprints on a site, or we want to take fingerprints from a person, but how would we do that? So we have chemical methods and powder methods for that. Both of them are basically dyes. So chemical methods include ninhydrin, a red color dye, sodium nitrate, a blue color dye. So what they do is they put the dye on the hand or the fingertips and take an impression of that on a paper. Similarly, it goes around with powder methods. They dab a small amount of black colored powder on your entire hand and then take the impression on a paper. So that is how you get these methods. One of which is also implied in a department, but we didn't get the chance to practice it. Now, although I have mentioned fingerprints are unique to everyone and serve an important role in forensics and biology, there are also some genetic disorders which lead to absence of fingerprints. So if we look at these genetic disorders, first one is A-dermatoglyphia. This is caused due to variation in SMARCAD1 gene, and it has been only reported in 5 extended families so far. I think out of which 2 are from Europe, the rest 3 are in America. And another other conditions that lead to absence of fingerprints are DPR, dermatopathia pigmentosa reticularis, and the second one is Naegeli-Franceschetti-Girardin syndrome. So if you look at the similarity in these two, one can't just define which one is which. Like DPR and Naegeli-Franceschetti-Girardin syndrome have so much in common that it is very hard to distinguish these two because they are caused by the same gene, that is keratin 14 gene. So the only difference in these two is that in the case of DPR, the pigmentation, like your fingertips, they become black or blue in color. That is very chronic. So you see, it is also accompanied with, you know, a lot of deformation in your teeth. But in the case of Naegeli syndrome, you don't have acute pigmentation of your fingertips and your teeth are perfectly normal. Then we have genodermatosis. This can also occur in areas other than the fingertips. So this is a cutaneous gland, disorder. So the cutaneous tissues of your skin, they get damaged in this. The next one is dyschromatosis universalis hereditaria. So if you break down the first word, you can easily conclude that this has something to do with the chromosome. So what happens in that in the double sequence of RNA, the first chromosome, there is an absence of an entire gene sequence of ADAR1, which leads to, just plain smooth surface on the fingertips. You don't even have a single ridge over there. And the last method to get rid of these fingerprints is surgical removal of fingerprints. So what happens is either they burn the fingertips so that the fingerprints can't regenerate. Because if you have observed that if you get a cut on your fingertip and when it heals, your fingerprint regenerates. But if your hand somehow gets burned and if you're burning the fingertip, the fingerprint won't be able to regenerate. And another method used in surgical removal is what they do is they take skin from some other parts of your body and then they just apply it, or you could say just cover the fingertips with that so the fingerprint just disappears. So here are some selected bibliography, you could say, for if you want to read more about fingerprint analysis and all. And at last, I have to talk about fingerprints in archaeology. So there has been one instance where fingerprints had been used in evolutionary biology of humans. So while some scientists were tracing out that why do we need fingerprints, they found that the pattern, or like we have arcs, holes, and loops. So this pattern is very similar in case of chimpanzees and orangutans. So that gave a huge support at the time of 1900 something to the fact that we have evolved from primates into humans. Also, using this fingerprints as a tool in archaeology has further scopes to it. So I think that is a field that we can explore more. So with this, we come to an end. If you have any questions or you would like me to elaborate on something, if I can, I will help you with that. So thank you.